And the next speaker will talk about the way to eliminate antibiotics from protein production. Please welcome co-founder and Chief Executive Officer of Gearbox, Biosciences, Dr. Arvi Jõers.
Yes. Hello, everyone. Oh, sorry. Okay. I'm Arvi. Nice to see you all, and thank for the organizers for asking me to speak about what we do at Gearbox. So we are a Tartu University spin-off company, and we deal with protein production. And it's not the protein production that you heard already today, where you have a biomass and you produce proteins for food and feed. But these, I'm talking about recombinant proteins, which are specific proteins for one particular functions. And for these kind of proteins, you can use several different organisms, but the one that we are working with is E. coli. Of course, a very well-known model organism in a lab. And this is one of the main hosts for recombinant protein production. And these recombinant proteins are all different kinds. For example, you can have biopharmaceuticals, the drugs that are quite often produced in E. coli. For example, insulin, everybody knows insulin. This is a protein produced in E. coli. And also the hottest drug in the market right now, the semaglutide, the anti-obesity drug from Novo Nordisk. This is a bacterially produced protein as well. Also, all kinds of industrial enzymes can be produced in E. coli and are produced. And several proteins that are needed for this kind of new foods. For example, if we take seriously a lab-grown meat, then you need a lot of growth factors to make those meat cells to grow in the bioreactors, and these growth factors are also produced in bacteria. Okay, and this is just an illustration of biopharmaceuticals, and although it's dominated by mammalian cells because of antibodies, then E. coli is still a good, almost a quarter of proteins are produced in E. coli. Now, this traditionally protein production in bacteria have a little dirty secret, and this is that the antibiotics are used in the production process. And this is to ensure the highly productive process, and for molecular biologists, this is for plasmid selection, of course. Anybody who has ever worked with E. coli and recombinant proteins knows that you need to use antibiotics for selection. This is very convenient in a small scale, in a research lab, but in a larger scales, this becomes more and more of the problem. And this is the problem is that at first that those antibiotics now are used outside of medicine, and there is a sort of general understanding that the more antibiotics are used, the more widespread the antibiotic resistance is. And this is, of course, a very bad thing if your next pathogen is actually resistant to antibiotics. And to make matters worse, your production strains are actually antibiotic-resistant bacteria, meaning that they are actually carrying a resistant gene. And now this is a kind of another big threat is that this resistant gene can actually escape your bacteria, although it's done in a confined conditions, and spread in the environment to another bacteria. And of course, every now and then, this is a well-recognized problem, and every now and then you can see these kind of gloomy headlines in the news. So we should care and not use so much antibiotics outside of medicine. But there are very specific agencies that's job is actually to care. And these are, of course, these guys, Food and Drug Administration in US, and EMA in Europe, also European Food and Safety Authority. And we should pay attention because they make the rules that we have to live by. Whatever you do in the lab, if you want to go to the market with that, you probably will need a green light from regulatory agencies, and these are the agencies. And they do make those rules. And as a result of this, there are additional costs for the companies if you are using antibiotics in your production process. First of all, you need really to purify your end product and test that that all the antibiotic residues are removed in the final product. I mean, if you want to inject something to the human, it needs to be really pure, and the same applies to the food. And by definition, some batches will fail. That's inevitable. Then there is a problem of the waste management. And it becomes, because their environmental rules are more and more stricter, then the waste management is more and more expensive, and it actually consumes a lot of energy. According to one farmer representative, what's happening right now is that if you ever have antibiotics in the process, you basically have to burn everything. And if you have a 50-ton bioreactor running once in a week, then it's a lot of material and a lot of energy that goes into that. Then there are kind of secondary effects. There are more indirect costs associated. According to one pilot plant manager who I spoke to, he said that they can't take antibiotic consuming processes anymore because their insurance becomes very expensive. It's all fine if everything goes well, but when there is an accident and you spill a lot of antibiotic-containing liquid, then cleaning that up is so expensive that the insurance doesn't cover antibiotic-containing processes anymore. And of course, there is also an image problem. How can you be a green company if you use tons and tons of antibiotics? And this is a real issue that I was not aware of, but one of the prospects actually said that this is one of their main motivations to go without antibiotics. All right, so if it really is such a problem, then there surely must be already some solutions on the market. And there are. There are broadly two types of solutions for that. And now I'm going to a little bit more detailed into the molecular biology. The one is that you take a gene of interest and integrate into the bacterial genome. This is a tried and trusted solution, but it's very slow because you have to integrate multiple copies to get any meaningful expression level of the protein. So definitely more than 10, and you can't do it in E. coli, you can't do it in a tandem. You have to do it like one by one, and that takes months. And the other one is that you still use plasmid, but you use a alternative selection method. There are several. Usually it's a sort of gene complementation based, but it's not an ideal. Several companies are using these, but it sets all kinds of different restrictions on the production process, which then becomes more expensive, and companies don't like that very much. So as a result, there are still many companies that are using antibiotics in a truly large-scale production. Not so much in the food space, but definitely in the pharma space. Okay, so now we have come up with a new technology that we hope will be a real hit on the market. We call it the pop-out plasmid technology, or POP for short. And this is our technology combines the speed of making a new strains, and it doesn't need any kind of selection. And now I'm gonna tell you briefly how it works. So you take a gene of interest, put it into the E. coli chromosome. And not in a random place, but very specific landing pad that we have created, which is very close to OREC, the origin of replication of the chromosome of E. coli. And now in this state, your protein is not expressed. Cells grow nicely as wild-type cells, as long as you keep them at 30 degrees or lower. And now when you want to, and this is also a state where what's happening in a bioreactor during your increase of biomass, during your growth stage. Now if you want to start producing the protein, you only need to shift the temperature in the bioreactor for a couple of hours. And what happens is that specific enzymes, recombinases, are cutting out this piece, and this now becomes a separate DNA molecule, a plasmid in the cells. And this plasmid starts to replicate. And now if you put these two things together, this gives you a selection-free production. So when the cells grow, your gene of interest is still a part of the chromosome, and it cannot be lost. This is a very stable configuration. And once you do the switch and the plasmid is formed, it starts to multiply, the copy number will go up. But because you also remove OREC from the chromosome, these cells do not divide anymore. And because plasmids can be lost only from dividing cells, these plasmids that are formed stay inside the cells, and they are just used as a template for protein production. All right. This is just an example that indeed we can produce several different proteins in the system. And I think this is something that will take over bacterial protein production in the coming years, so that antibiotics are sort of pushed out of this space for several reasons. At first, regulations will get tougher, not easier. There is no way that tomorrow FDA will come that, we don't care about antibiotics anymore, you can do what you want. No, that will not happen. Then there will be more technologies available that enable antibiotic-free production. Whether our technology will win or somebody else, I don't know. Only time will tell. But there will be more options in the market. So it will be easier to switch. And there will be a bigger market available because more and more proteins will be produced. I think this semaglutide from Novo Nordisk is just the first example of something that is branded as a drug, but actually it is perhaps more than just that. It's also a lifestyle drug or product a little bit. And I'm pretty sure that more like these will be coming. Yes, and that's my last slide, and thank you for your attention.
Questions?
Yeah, so I didn't get it. So if you do the antibiotic in dependent production right now, let's say batch type production in reactor, you finish the production cycle, the waste solution that contains antibiotics. Do you have to clean it or not before you pump it into sewer?
Oh, definitely, yes. You have to clean it.
Everything needs to be taken out?
Yes. Depending on the antibiotic, what you have to do is different. I know, for example, for kanamycin, the sort of technical procedure for inactivation is that you have to raise the pH to 11 and then you autoclave it. All the solid waste, as I said, has to be burned.
Okay, so it actually doesn't contribute to this kind of antibiotic, or proliferation of antibiotic and all the negative aspects, the link to that you showed in your first slides?
Well, it does, in a sense that it's all these kind of processes are never like 100%, and there is always a risk of spill. There is a it's been sort of well documented that that increasing the probability of these kind of things actually increase the probability of finding antibiotic-resistant bacteria in surrounding environment. So yes, if everything goes like 100% as planned, then perhaps it doesn't leave the plant, but in reality it's not so.
So do we have more questions?
But this technology, as far as I understand, is applicable only in case of bacteria.
Yes, we have built it only in E. coli right now. In principle, it could be also used in other bacteria, but we haven't tried.
Okay.
Yes, please.
Thank you. Your talk was crystal clear, and it's a super interesting tool. You did mention in introduction that it was a spin-off from academic research, and I'm curious in understanding how did you find out? Like, was it were you researching homologous recombination or defense mechanisms or effect of temperature? How did the idea come about?
Oh, that's a long story. Do you have 30 minutes? No. Well, it was like a gradual process. We started a completely different end in mind. So we started working on the sort of technical aspects of sort of removing OREC from the chromosome and stop bacterial growth. And then it sort of gradually developed more towards the industry application. We took the patent, then we realized that if we want to really to sort of get it off the ground, we have to start the company as well, and so it builds on that.
And very quick one, how long can the plasmid be? That can recombine.
Like here?
Yeah.
Well, because these cells are actually not dividing anymore. It's like a terminally differentiated cells. So as long as the cell lives, the plasmid is there, yeah, and it produces protein. We know that they can produce proteins for a couple of days, but then we haven't run very long experiments because this you have to do in a bioreactor in the fed batch conditions, in special conditions, and we haven't done it yet.
Thank you.
Days.
Yeah, thank you. And more questions? So then, thank you for your nice presentation.